Thank you very much. So that was our introduction, and it's great to finish it with the young people who are really the centre of everything we do. So moving on, we're going to move to our tissue engineering and regenerative medicine theme. So it's a pleasure to introduce Professor Jane Sowden. I'm looking around the room, it's over there. Yeah, please come on. Jane is Professor of Developmental Biology and Genetics at the UCL Great Ormond Street Institute of Child Health, and she's co-lead of the BRC's Tissue Engineering and Regenerative Medicine theme.
Thank you. I'm delighted to introduce the Tissue Engineering and Regenerative Medicine theme to you today. So as Laura said, I'm Jane Sowden. I co-lead the theme alongside Professor Paolo De Coppi, supported by our junior faculty at Gosh and Alderhay, our partner site, and also by our deputy theme leaders and our research manager. So the theme is aiming to develop new treatments for children who are born with structural malformations and tissue failure. And these are major causes of infant mortality and disability. At Gosh, each year, around 40% of children who visit are affected by a birth defect, and for many of them, there are few effective treatments available. So there's an unmet need for new treatments to try and replace missing, damaged or undeveloped tissues and organs, and for personalised devices and precision surgical procedures. So the theme is working to try to improve the life expectancy and quality of life for children by pioneering the repair and reconstruction of tissues and organs. So our approach is to bring together expertise in stem cell biology, in tissue engineering, and in surgical sciences. So we're using the stem cells and scaffolds to conduct proof of concept studies to grow tissues for reconstruction, as well as using these models to study pathology of disease. We're progressing preclinical studies to grow GMP-compliant tissues and organs that are ready for transplantation, and we're also undertaking first-in-child studies and developing new surgical tools that will improve outcomes for children undergoing surgery. And so overarching this work is our flagship surgeon scientist training program, where we're supporting, together with our partners Alderhay and also Gosh Children's Charity, a program for surgical trainees to undertake pre-doctoral and also PhD training. And this is helping to train the next generation of research leaders who are surgeon scientists. And of course, across all of this, we're working closely with patients and families, and also with the charities who represent them, to make sure our research aligns with patient priorities. So what I'm going to do in the next 3 slides is highlight some of the work along the pipeline before I introduce 2 of our early career researchers who will give talks on 2 aspects of the theme's work on growing 3D tissue and in surgical innovation. So first then, using our organoid. So first of all, the work from Paolo De Coppi and Mattia Gurli's labs have developed new tools for fetal diagnosis. So they've been able to show that stem cells can be isolated from the amniotic fluid surrounding a baby in the late stages of pregnancy. And these stem cells can be grown in the lab to create miniature 3D organs, which we call organoids, such as the lung or the intestine. And these represent the baby's biology and genetic profile and can aid diagnosis and also help to understand why some tissues don't develop normally. And one condition that the team have studied using organoids is the condition congenital diaphragmatic hernia, where a hole in the diaphragm leads to pressure on the developing lung, impairing its growth. And the team have been able to mimic this impaired growth due to the compression in the lab and test a novel therapy for CDH that promotes lung growth. And then in our work towards developing tissue-engineered products ready to restore damaged tissue, our teams are focusing on conditions affecting the esophagus, the intestine, the thymus, and the retina. And for patients whose sight is impaired due to retinal dystrophy, we're developing retinal patches in which light-sensing photoreceptors are grown on tiny scaffolds, and these cone and rod photoreceptors are isolated from organoids grown from stem cells in the lab. And the scaffolds are designed to be inserted into the subretinal space at the back of the eye to repair tissue and restore sight. So in work led by Stefano Giuliani, we're developing fluorescent dyes to improve diagnosis and guide the precision of surgery. And so in the condition necrotizing encephalitis, which is a life-threatening condition where the intestine becomes inflamed and dies, using fluorescent dyes that label, shown in green, the healthy perfused region of the intestine, the areas that are diseased and that need to be removed can be more precisely defined. And the team are developing a similar approach for targeting tumours in neuroblastoma, and so again to enhance the safety and precision of surgery to remove tumours. And so that's looking ahead, where we'll continue to work with patients and families to engage them in discussions about the new therapies, and also gathering input for clinical trial design. And we're continuing to build our networks and collaborations in order to develop new tools and new treatments that will transform the care of patients with tissue malformation and organ failure. And so that leads me to introduce our term early career researchers. First of all, we're going to have a talk from Giovanni Giobbi, who is a senior research fellow, and he's going to talk about some of the work building mini stomachs in the lab to understand gut health and disease. And then that will be followed by Aaron Keeley, who is one of our surgeon scientist PhD fellows, who is going to talk about treating spina bifida before birth using a minimally invasive operation. So before I welcome them, we're going to take questions for all three of us at the end of these next two talks. So thank you, and I'll hand over to Giovanni.
So thank you. It's a pleasure to present one of the projects of the tissue engineering theme. This project is focusing on building mini stomachs in the lab to understand gut health and disease. The problem that we are trying to address here is that 1 out of 200 British children are affected by different stomach conditions. These span from common issues such as gastroesophageal reflux disease to gastritis to more rare genetic conditions that have currently no models available, no cure to date. So the goal of these particular projects is to grow mini stomachs in the lab that are derived from patient organoids. These organoids can assemble into more complex assembloids, and these assembloids resemble a mini stomach, so they are able to model and treat gastric pediatric disease in a dish. So the particular problem we're trying to address here is one rare genetic condition, is a congenital disorder of glycosylation caused by a particular gene, the PMM2. When this defect in the gene is associated with another genetic mutation, we have the antral foveolar hyperplasia. So it's a gastric manifestation of this disease, where we have lumps, overgrowth of cells that are defined as polyps in one area of the stomach. So the stomach presents inflammation, and these kids have difficulties in digestion, in production of the gastric acid in the stomach. So the goal here is to build mini stomach assembloids from this particular cohort of patients to model the antral foveolar hyperplasia in vitro that have no other animal model available. So we are building the first human-specific in vitro model for this. So through endoscopies, we are isolating tiny biopsies from the patients from the 3 areas of the stomach and deriving organoids from the stem cells of the epithelium of the stomach. So the human stomach is composed of 3 main areas, but if we derive organoids from the 3 areas of the stomachs, then these organoids are not cross-communicating. There is no crosstalk between the 3 areas of the stomachs. But what happens if we assemble the 3 areas back again into a complex assembloid system? Will this system be able to model efficiently the disease that we are investigating? So we'll present here just very few results. So these are 2 assembloids, one from a healthy patient and one from the diseased patient with a PMM2 mutation. As you can see, the lumen of the inner part of the assembloid of the healthy patients presents no lump of cells, no overgrowth, no polyps. While in the antrum specific area of the assembloid derived from the patients, we have overgrowth of cells, so these polyps are present. Therefore, we are able to model efficiently the antral foveolar hyperplasia in a dish. What is happening to these patients? Why they have such difficulties, such high inflammation? So this is one of the functions that is mostly affected. As you can see here in the healthy assembloid, we have functional acid production in the fundus. This acid production is responsive to drugs, while in the diseased patients, we have no acid secretion that is actually quantifiable in our in vitro system. So something is happening. There is a function that is off in these patients. So what we're doing now, we're trying to test drugs in our in vitro model. First, we tested a repurposed drug. The eparestat was a drug used for glycemic issues in diabetic patients. We know that it increases the glucose available in the bloodstream, and this can help this disease, particular mutation, the PMM2, to function well. So if we repurpose this drug, are we able to recover the phenotype, to arrest the overgrowth of polyps in the stomach? And you can see here, we are able to decrease the level of proliferation in the antrum of the assembloids by giving this drug. So we can revert the phenotype in these patients. Same thing applies for the mannose treatment. It's a drug that functions in a different way, but with the same output. And also in this case, we are able to decrease the proliferation in the antrum. So these tell us that we can actually model in vitro this disease. The future research, we will be focusing with this assembloid. We will go from the stomach specific to, we will broaden the application to gastroesophageal reflux disease in the upper gastrointestinal tract by putting together more organs, so the stomach, the esophagus, and the lung, to explore the early onset of gastroesophageal reflux disease. So what happens to the cells in the esophagus and in our lungs when we have an overproduction of gastric acid in our stomach? So this is what we're trying to address now, and this is the team behind the work that I just presented. So I'll leave the stage now to Arun. Thank you.
Perfect. Thank you, Giovanni. Unlike Giovanni, I'm a pediatric surgeon, and I'm grateful to the BRC for the opportunity to do this research and to be part of the NHS's only commissioned service for fetal surgery for this condition, which is a team effort between Great Ormond Street, UCLH, and a big Belgian centre in UZ Leuven. There's a bit more background to my talk, so apologies for that, but hopefully it will be a good story. So we all know that spina bifida is the most common congenital anomaly of the central nervous system. And despite our knowledge of the protective effects of folic acid supplementation, it still has a relatively high incidence of 1 in 2,000 across Europe. It arises due to incomplete closure of this structure called the neural tube, which happens in the 4th week of gestation. In the bottom right, you can see a normally folded and closed neural tube, which will go on to form our spinal cord. And above that is a section of open neural tube, which, if it remains like that, will become spina bifida. Now, I'm focused on the most severe type of this condition, which is termed myelomeningocele, where the exposed spinal cord is on the surface of the back. And we believe that a combination of this abnormal development, together with chemical injury from the amniotic fluid, as well as mechanical trauma against the uterine wall, which is termed the 2-hit hypothesis, results in progressive nerve damage throughout gestation. Affected children, therefore, have a range of significant challenges which require lifelong medical care. Now, this observation that there's a sort of second hit, that throughout pregnancy there is loss of nerves, led to efforts to cover the spinal cord before birth in the womb. And it turned out that if you did do that, you could rescue nerve function and improve the status of the babies at birth and onwards. And that resulted in this landmark randomized controlled trial, which compared open fetal surgery before 26 weeks of gestation to standard postnatal repair, which happens in the first few days after birth. And you can see in the image on the right that this was via an open approach, so a large incision in the uterus of about 6 to 8 centimeters. These are the main results, and one of the headline findings was if you had a fetal operation, you, at 2.5 years of age, nearly twice as many children were walking independently. And that felt like quite a big win, understandably. But if you look lower down that chart, you can see that the open approach was associated with a couple of major problems. So for 1 in 10 of the mothers, the uterine wound did not heal properly, that's termed dehiscence, and 8 out of 10 babies were born prematurely. And so all across the world, centers thought, what's a better way of achieving the same thing? And they came up with this more minimally invasive fetoscopic technique, which uses 2 or 3 5-millimeter incisions in the uterus to try to perform the same operation. The problem that we are currently faced with, and the question my project is trying to answer, is this fancy operation better or even equivalent to the now gold standard open fetal operation? And the reason that's even a question is that everyone is doing a slightly different thing. There's variation in patient selection, surgical technique, and the outcomes that centers are reporting. So this is my plan, which is missing some words, sadly. Anyway, I'll touch on the, the systematic review and meta-analysis, looking at the outcomes of both open and fetoscopic repair, as well as the picture on your far right, which is to do with the use of computer vision, which is a subdomain of AI, to work towards automated analysis of the videos of fetoscopic surgery, so the computer can tell us how we're doing. So if you have a look at the top of the screen, you can see that at the moment in the world, there are 4 different ways of accessing the uterus to perform this operation. That's just one of the many factors that varies between centers and between patients. And I began by looking at the proportion of studies that reported important outcomes. So you can see for the mother, I've put 3 of the key complications that can happen after fetal surgery, and then a couple of the key outcomes for the baby, which reflect an unsuccessful fetal operation. And I think you can see that the outcome reporting is both selective and inadequate. So we don't really know what the outcomes are. And if we then move to just a little bit of the meta-analysis, it also is clear that, well, it appears that the outcomes vary between different techniques. So if I draw your attention to the graph on your right, if you look at revision of repair site, what that refers to is an operation performed after birth to revise the operation that was performed in the fetus. And it appears that that is more common if you have the fetoscopic operation as opposed to the slightly more old-fashioned open operation. So this is sort of the background to why this work is important, because we should be aiming to standardize and provide the best procedure and care for the mother and baby. And to that end, we've established an international consortium of 15 centers across Europe, North, Central, and South America that perform both of these techniques, if you like. And this is for the two sort of middle pictures that you saw briefly of my plan, which is for a consensus process, which is going to happen in the next couple of months, to try to bring the world closer together in terms of how they should be performing this fetoscopic operation, and also to perform a multi-center cohort study so we can, again, try to move closer to understanding the real outcomes of the fetoscopic operation versus the open. And finally, perhaps the most innovative and exciting bit for me is the computer vision piece. So this sadly takes hours of my life, but what it involves is working towards training a model to initially recognize the different phases and steps of the operation, which we call surgical workflow analysis. There are lots of uses for that. It can be helpful to allow us to monitor our performance over time and also detect deviation from the normal sequence of events. In other words, before or even when a complication arises. You can even do more fine-grained analysis, which takes even more time, which involves training the model to detect the instruments that are coming in and out of the view and the tissues that they're interacting with. And that results in the computer seeing something that looks a bit more like this. And that can allow us to have a much more detailed assessment of what's actually going on. And the ultimate aim of this piece of work, which the machine learning engineers tell me will take years, is to try to correlate what is happening in the operation with the patient outcomes in the years that follow. Thank you very much.
Thank you very much. If I can invite Jane and Giovanni to come back up. We have a few minutes for questions now. Remember, online you can ask your questions through Slido, and there'll be roving mics in the room as well for anybody in here. So would anybody like to start? I've got a question online to begin with. So, David.
So can you hear me? Yeah, so you got 4 questions online. One speaks to the introduction by Thomas. Does each theme have a group of children and young people advising them or working with them?
And that's probably for Jane. Oh, Thomas.
So all the themes are working with different patient groups, and the young person groups are overarching and are working with all the themes. So it's not a one-to-one allocation, but it's much broader than that. And all the themes will also work a lot with individual charities and their patients according to the different diseases they're working on.
Thank you very much. I can't see any hands in here, so we'll go to the second question online.
So we've got 3 questions so far, one each. Jane, this one's probably for you. What are the biggest challenges in translating tissue-engineered products from lab to bedside?
Thank you for the question. So the question is about translation from the lab to the clinic. And I think that this is a big challenge. And of the projects that I've outlined, many of those have managed to lever additional support from additional funders to take the proof of concept work in the lab into animal models. So for instance, for the esophageal engineering, the retinal tissue engineering, that's one of the things we're doing. And another challenge is to make those clinical grade products, which requires a tech transfer, if you like, from the laboratories, our research laboratories, into clinically ready approved products.
Next question online.
Well, it appears that there's an avalanche of questions coming through online. Next one to Giovanni. Do you plan to include other cell types in your models?
So for now, it's this one is focusing on the epithelium, but we are already including fibroblasts, so the mesenchyme, especially when putting together multiple organs. They don't assemble so easily if they are epithelium only, so we have another layer of cells to be added, which is the fibroblast, and then we're planning to add the endothelium, and eventually, with the evolution of the project, an immunocompartment for specific diseases in the future.
If I just stick with you there. There's a follow-up, well, it's not a follow-up question, but it's for you. How big is the leap between the mini stomachs in the lab and in vivo applications?
So this is what we are trying to address and to reduce, because the leap from simpler organoid models is quite big. We are working in increasing the complexity to cover this gap and have a more functional system. So I do believe that we have a very good increase in function with this increasing complexity. For example, the acid secretion is not present in the organoids alone. So we already have one function more, so we are getting more and more closer by enabling the crosstalk between the different cell types of the different areas. So yeah, it's a good system to work on.
Thank you very much. Just checking if there are any questions in the room. No, if not, oh, there's one hand further up. We're just going to take one in the room next.
Hi, a question for Aaron. I was wondering, do you think if you train the software up well enough, it will be able to assist sort of during the surgery as well instead of afterwards as well?
Yeah, it's a really good question. So you're absolutely right. One of the potential indications is a decision support system. And I think particularly in an environment where, let's say, the team is not as experienced, it could really help in real time, and people are already working on this, to highlight, like I mentioned, a deviation from perhaps what is a more normal sequence of steps, and also in appearance. So you're absolutely right. That is what everyone is writing will be one of the applications of this technology.
Thank you. We'll take one more question online.
Again, for you, Aaron, slightly unfair, but how do you hope to influence other surgeons in the way that surgery is performed across the world?
Yeah, I think that might be the biggest challenge of what I described. And perhaps that's because it's a folly to believe that there is a better or a even best approach. That probably isn't true. I think the way to change opinion and perhaps convince people is with high quality data. And that's the main thrust of what we're trying to do. Because at the moment, if you, as I have had to do, if you look at what's been published, the vast majority of it is single centers publishing their own experience over time, which is improving and is very useful. But the inherent biases in that way of reporting data, I think, really limit its meaning. So hopefully, with that map you saw, and by bringing people together to contribute their data, we will be able to move towards something better.
Thank you very much. In the interests of time, I'm going to say thank you once again to all three of our speakers here.